Hi everyone, welcome to Medical Microbiology. So today we will discuss chapter 25, which is RNA viruses that infect human. On the first lecture, we will discuss the first group, which is the segmented single-stranded RNA viruses. Okay, compared to the DNA viruses, the RNA viruses that infect human is actually more diverse. It means that there are more type of RNA viruses that infect human compared to the DNA viruses that infect human. So these are the list of several RNA viruses that infect human. The most common one, as we know, so this is the one that causing infection almost every year, which is the influenza viruses. There is another viruses called the hantaviruses, parainfluenza, mumps viruses, morbilliviruses, respiratory syncytial virus, rabies, SARS, this including the COVID-19. This is coronaviruses, ruby viruses, hep C viruses, St. Louis encephalitis virus, yellow fever, dengue fever, including Zika fever over here. And then HIV, the one that causing AIDS, polio, the one that causing poliomyelitis, Hep A, rhinoviruses, norovirus, rotavirus, Ebola. And the last discussion will be about the prions, which is just a proteins. It's not really virus, but is unfolded protein that cause infection to human, especially in human brain. Now we go to the type of RNA viruses that infect human. It is really similar with the classification of DNA viruses. Remember, human is considered as animal. Therefore, this virus will be considered as animal viruses, which is what it will mostly have the envelope. Animal virus mostly have envelope. So it has capsid, and then the envelope, and of course there will be spike on the envelope that will be used for attachment. And inside the capsid there will be the nucleic acid. In this case will be RNA. So RNA. So these the envelope viruses. And just remember, because mostly the RNA is single-stranded, therefore the left side over here, the most common type will be the single-stranded RNA, ssRNA. Okay, and some of the RNA is actually segmented. Segmented means more than one segment. So like this one over here, this one has two segments. RNA. Non-segmented is the one that only carry one segment of RNA. So if we compare, it's gonna be look like this. So this one, the non-segmented, will only have one single-stranded RNA, and the other one over here, the segmented, might be two, maybe four, maybe sometimes eight. There are eight segments. Okay, so this is segmented, and the other one is non-segmented. So this is segmented. This is non-segmented single-stranded RNA. Okay. So again, this will be, the left side over here will be the most common type of RNA viruses that infect human, which is the one that have envelope, but including this SIV over here, the one that have envelope and carry single-stranded RNA. The other one over here will be the one that do not have envelope. This is the naked viruses. And the most common of these naked viruses will be the one that carry single-stranded RNA. So this is ssRNA. And the least common one will be the one that carry double-stranded RNA. Okay, so this one gonna be like this, double-stranded RNA. And this one will be just single-stranded RNA. No envelope, because this was this naked naked viruses or non-envelope viruses. Okay, so for lecture 1, chapter 25, we will focus in this one first, okay, which is the envelope segmented single-stranded RNA. There are 3 groups, okay, flu viruses or orthomyxoviruses, and then Bunia viruses and RNA viruses. So this is going to be our discussion, our topic for lecture 1, chapter 25. Okay, so it's going to be the envelope segmented single-stranded RNA viruses. So if we make the picture, it's going to be this capsid, then this envelope, spike, and the envelope for attachment, and the single-stranded RNA. There are more than one segment. Okay, so that's going to be how the structure of this virus, okay, going to be envelope segmented single-stranded RNA. Again, the first one, flu viruses or orthomyxoviruses, Bunia viruses, and the RNA viruses. We start with the first one, which is the orthomyxoviruses, or the flu or influenza viruses. This is the most common viruses that attack human. Every year there will be flu, there will be influenza, and every year there will be flu shot, different flu shot. Because what? Because this influenza virus, they actually can change their spike, and they also can substitute their RNA segment. Therefore, almost every year there will be different subtype of influenza. So the flu shot that we have last year maybe will not be effective for this year. The one that we have this year, maybe it will not, really effective for the next year, because the flu is always changing. But it is better to have the flu shot every year, in order to protect us from the flu that may be spread during that year. Now, the most common one and the most dangerous one is type A, type A flu viruses. And this type A actually have different subtype based on the spike. So the spike, which is the H spike and the N spike. So the spike for the virus, the flu virus, it has 2 subtype, which is H spike, okay, and the N spike. Now the H spike is actually, there are H1 until H15. So there are 15 subtype of H spike. The N spike, it has 9 types, so 1 until 9. That's the reason why it has number over here. So H1N1 is mean that it has subtype 1 for the H and subtype 1 for N1. And this is considered as the most dangerous type of flu viruses. And this is the one that kill millions of people in 1918, 1919, which is 100 years ago. It's called the Spanish flu. Right? And then it's recurrent 76, 77, 91, and 2009. So this is the most dangerous type of flu viruses, flu A. And then H2N2, it's appear mostly in Asian pandemic, Southeast Asia, and then in China, H3N2 in 68 and 94 in Africa. So these are the type of A viruses that's considered very dangerous to human. Okay? And then B and C, they are less virulent or less dangerous compared to the type A flu viruses. Now, how they infect human? And remember, there are 6 stages of cycle of viruses. And because this is animal viruses, then there will be spike, and spike will be used for attachment. So this is going to be the first stage, which is attachment. Okay, the spike will attach to the respiratory tract cell. So therefore, the most common site for infection will be the nose. Okay, because nose is part of the respiratory system, respiratory tract. So attachment to the cell on the respiratory tract organs. And then after that, the second stage will be the penetration. And the RNA of viruses will be released into the cytoplasm. There are 8 segments. Remember, this is segmented RNA. So there are 8 segments of RNA that are carried by flu viruses. And this 8 actually is negative sense. Remember the difference between negative sense and positive sense of RNA. The negative sense cannot be used for making protein. Therefore, this negative sense of RNA will be entering into the nucleus of the host cell. Okay, inside the nucleus, this negative sense will be converted into positive sense. So this one is positive sense. It will go out from nucleus into the cytoplasm. And then they will use our ribosome, the host ribosome, to make the spike. So the spike then will be attached into the host cell membrane, into our cell membrane of the respiratory tract cell. And then inside the nucleus, the positive sense over here will be also used to replicate into negative sense of these RNA viruses, the RNA segment over here. And then this RNA segment will be out, million of them actually, and then every 8 of them will assembly. So this is the next step going to be assembly, okay? And then after that, the last step will be released from the cell. When it is releasing from the cell, it will carry the cell membrane of the host cell and the spike over here. So therefore, the envelope, remember, the envelope of animal virus is actually part of the cell membrane of the host cell. And flu virus is causing what we call the lytic or acute cycle or infection. It means that they actually kill our respiratory tract cell very fast. Within 7, 5, 10 days, they actually destroy a lot of our respiratory tract cell. And therefore, if our immune system unable to take care of this infection, then mostly, the patient will be die. That's what happened with the Spanish flu that killed millions of people, okay, because of the destruction of respiratory tract cell by these flu viruses. Okay, so this is a lytic or acute type of infection, really fast type of infection. Usually, within 10 days, if our immune system able to uh, defend our system against this virus, then we'll be cured, after 10 or 14 days of sickness or illness or flu. Okay. And again, the key of the influenza or flu infection is this spike over here. So the spike. And there are two types of spike, H spike, and there are 15 of H subtype, okay, 1 until 15. And then the N spike, there are 9 subtype, so 1 until 9. Okay, so therefore, usually the flu virus sometimes will be named based on what type of spike that they carry. So like H1, N1, H2, N2, H1, N5, H2, N9 maybe. Okay, therefore there will be a lot of type of flu virus actually, just based on the spike that they carry. And this spike, let's see over here. This is the H spike, the blue one, and this is the N spike, the brownish one. So they have different structure, and they can be changed. So maybe this is H1, this is N1. Maybe this is H2, this is N5, for example. Okay, so they're able to change this spike every year. There's the reason why the flu shot for this year will not be effective for the next year. The flu shot that we have last year maybe will not be effective for this year. Therefore, we have to have or to get flu shot every year because of this changings or drift of the spike every year. Okay, so again, that's what we call the mutation. So this virus, the flu virus, is actually mutated every year. They can do what we call the antigenic or antigenic drift, which is changing the H, the spike, the H and N spike over here. They change it. They can change it. So therefore, it's going to be different virus this year compared to last year, and then there will be maybe next year will be different type of virus just based on the spike. This is what we call the antigenic drift. The second mutation is called the antigenic shift. It means that they actually able to shift or change the RNA from one virus to another virus. For example, here, this is a bird flu viruses. This is the avian viruses, which is the one that attack bird, and this is human viruses. There are two viruses over here, bird viruses and human flu viruses. And then they actually able to infect the same animal. Let's say this is swine. Okay, and then inside this swine, then what happened? They able to change maybe one or two segment. Like for example, the blue segment is coming from the bird, and the red segment is from human. And then just one blue enter into the human viruses. What happened? It's become new viruses compared to these viruses. This is already different type of viruses. Therefore, if, for example, we already immune against these viruses, because we have disease or we have flu shot for these viruses, and on the next year, maybe this is the virus that infect human. What happened? Then we will not have the immunity against these new viruses over here. Then we will be ill. We will have the sickness from this type of viruses. So just remember, the flu viruses, they change, they mutated every year, very, very fast. Okay, again, there are three main strains, and the most dangerous one or the most virulent one is type A, and this is the one that have killed a lot of people uh, lately. So especially older people, they are more high, have a higher risk of getting this flu A infection. Therefore, it is required for older people to get the flu shot every year in order to protect them against this influenza A viruses. Influenza B and C, this is considered as less virulent or less dangerous compared to type A. Okay. Again, type A is highly acute. Right? Remember, this is acute type of infection and highly contagious respiratory infection. This is seasonal and pandemic. It means that it can be transferred across the states, across the countries. Right? We know the sign and symptom for these viruses. As a diagnosis using some immunologics tests, treatment mostly using some flu drugs, okay, like maybe Tamiflu for H1N1, okay, Relenza, and vaccine is usually available every year. Okay. The second type of segmented ssRNA envelope ssRNA virus is Bunya viruses, and this is a kind of endemic. It means that's a local infection. It's not really pandemic. Several types like hantavirus and Sin-Nombre viruses is considered as the most dangerous type of these Bunya viruses, and usually it's transmitted by insect, like insect, like a mosquito and ticks. The last one will be the RNA viruses. Okay, this is also mostly pandemic. I mean, endemic in South America, like Argentina, Bolivia. It can cause hemorrhagic fever. Hemorrhagic fever means the patient will have a high body temperature and sometimes with bleeding, accompanied with bleeding. So it's kind of type of dangerous viruses also. I think that's all for the first lecture, the segmented SS single-stranded RNA viruses. On the next lecture, we will discuss the non-segmented single-stranded RNA. Goodbye now.